alaikum. Today's article is titled Efficacy of Different Irrigation Activation Systems on Bacterial Extrusion, which was conducted by Turkish group and published in Australian Endodontics Journal in 2020. A couple of weeks ago, I presented an article that compared post-operative pain after using different irrigation protocols. In that article, the authors correlated the pain with the bacterial extrusion during the activation of irrigant. And in this article, the authors mentioned that during irrigation, vital or necrotic pulp fragments, bacteria, and their products can be extruded from the apical foramen and accessory foramina to periapical tissue. And the apical extrusion of infected smear is undesirable and is accepted as major cause of flare-ups and pain following endodontic treatment of infected teeth. So, the aim of this study was to compare the effect of different irrigation activation systems on the amount of apical extrusion of bacteria in root canals contaminated with Enterococcus fecalis. In this study was 90 single-rooted teeth with straight mature apices, and single canals with no tooth resorption were included in this study. The final irrigation was performed with 2 ml of 17% EDTA to remove the inorganic debris produced during root canal preparation and to open the dentinal tubules for bacterial growth. And the outer surface of the roots were covered with nail polish to prevent any microleakage through lateral canals during this procedure. And at this stage, no contamination was applied to 15 randomly selected, and they were separated as an active control group. And then 20 microliters of bacterial suspension were enucleated into each canal. And to confirm successful contamination with the E. fecalis, bacteriological sampling of all teeth was performed. To the scanning electron microscopy analysis, teeth of positive control and negative control groups were cut into 2 pieces using a chisel. And in the SEM images obtained after preparation steps, no bacterial growth was observed in the teeth of the negative control group, and bacterial colonization was observed in the root canals of the teeth of the positive control group. And for the collection of extruded bacteria, agarose gel was used to simulate periapical tissue and to collect the extruded bacteria. The entire root surface was then covered with the agar, and then agar was allowed to solidify for 1 hour. And after solidification, the construct was placed in a microcentrifuge tube and held there during the irrigation activation procedure. And for the irrigation activation, the samples were assigned to one of the 4 test groups, which are standard needle irrigation, EDI, endoactivation, and endovac. And for the microbiological analysis, after the irrigation activation procedures, the system devices were taken separately, and the micropipette tip containing the agarose gel and extruded bacteria was immersed in a tube with 2 ml sterile saline solution, and then the colony-forming units grown were counted. Irrigation activation with EDI resulted in significantly more bacterial extrusion than irrigation activation using endovac, but there was no significant difference between the positive control group, endovac, standard needle irrigation, and endoactivator systems in terms of bacterial extrusion. And similarly, there was no significant difference between EDI, SNI, and endoactivators in terms of bacterial extrusion. So to conclude, none of the irrigation systems tested in the present study prevented bacterial extrusion, but endovac is preferable to EDI as the former caused less bacterial extrusion.